is now going.
Okay. All right. Again, thanks to everyone who is here. We want to talk a little bit about the vaccine trials. This has some statistical content intended really for a broad audience of statisticians, clinicians, general interest regarding those trials. So there are currently 4 large ongoing vaccine trials that are making the news right now from AstraZeneca, Janssen, Moderna, and Pfizer. Pfizer's is also sponsored by BioNTech. All of these companies have made their protocols publicly available, and certainly want to say this is rarer than probably I would hope it would be. But the fact that these are publicly available now is fantastic, that we can go through and look at them and see the details and see everything that went into them and really kind of get a good idea of what's going on and have comfort with these. We all want to have a safe and effective vaccine, and we would like that as quickly as possible. This is an ongoing pandemic. And so to the extent that we can do this fast, we would like to do it without compromising safety or efficacy. General outline today, we're going to talk a little bit about what they're estimating in these trials, both from an efficacy standpoint, talking about vaccine efficacy, and also safety. And certainly keep in mind, I'm a statistician. I certainly am familiar with safety issues, but I'm not really qualified to get into the real nitty-gritty details of what kind of events. So I'm going to talk about them more in broad terms, short-term events, long-term events, but I'm not going to get into the details of this is the kind of illness that we might be worried about. So I won't have a detailed explanation of transverse myelitis, for example. We're going to talk a bit about general trial design, really aiming at the interim analyses, what the interim analyses are doing. Are there things that we ought to worry about with interims? And are there things that we don't need to worry about with interims? So talking about that, certainly some of this is my opinion, but I'm hoping a relatively informed opinion. And also, what kind of protections do these trials provide? So discussions of limiting the chance of false discovery, and also talking about the amount of information for different safety questions. So depending on what aspect of safety, you can have different levels of information. And that's going to be important for assessing what's going on with the interim analyses. So generally, all of these trials follow kind of a general vaccine trial paradigm. The goal of this is to vaccinate a large number of people. A large number can vary, but here it's 30,000 to 45,000. Actually, I think the Janssen trial is maybe 60,000. And these are going to be randomized. So some proportion get the placebo, some get the experimental vaccine. Often these are equally randomized. The AstraZeneca trial does 2 to 1. And then what we do is we want to measure efficacy and safety. And for efficacy, what we're going to do is we're just going to wait for infections to occur in the trial population. So we've got these 30 to 60,000 people. We're following them up, and we are continuing to observe them. And as events accrue, we're going to gradually wait for a certain number to happen. And of course, the goal in this is for a vaccine to prevent infections. So we're hoping for fewer infections on that experimental arm as compared to the placebo. If that is sufficiently strong, so we have a lot fewer infections on the experimental arm, then that would allow us to make an efficacy claim. And I'll get into the details of that in a moment. For safety, we tend to follow up several broad classes. Certainly, we're worried about immediate injection reactions and that might include the period going up to a week to a couple months in terms of what might happen. So there was in the 1976 flu vaccine had a number of Guillain-Barré incidents, for example. So we're interested in what happens to people right after they're vaccinated. All of these trials have a provision for long-term follow-up going out 2 years. And so this is for longer term events. These may be considerably rarer, but we want to make sure. And also be monitoring disease severity. So the notion of are we making the, we want to make sure that we're not making the disease worse, which paradoxically can happen with some vaccines. So we certainly want to monitor for that. So let's talk about efficacy. So vaccines certainly intended to prevent infection. Worth thinking about that within an individual. So you go and get your flu shot because you don't want the flu to be as bad, or preferably to avoid it. But this is also intended for across the population. So the ability to obtain herd immunity by vaccinating a sufficient amount of the population, and that protects everybody by limiting disease spread. Usually within a vaccine, we talk about something called VE, just vaccine efficacy, which is defined as the percentage of infections prevented. So if the vaccine efficacy is 75%, then that means that the vaccine has 1/4 as many infections as the placebo on average. Of course, there's variation in here. Currently, regulatory bodies are looking for an observed VE of about 50%. And in the context of the trials we're talking about, that's really demonstrating superiority to 30% VE. So remember, everything has noise associated with it. And so we're looking for about 50, and that will allow us to say, hey, we're definitely better than 30. Definitely being the usual statistical version. So other VE examples, just to keep in mind, just kind of written out a little table here. So if you haven't done anything to stop infections, you might be getting 1% of the placebo patients infected. And if you haven't done anything, 1% of the vaccine subjects are also going to be infected. So we can think about a relative risk. That would be about 1. So you have the same risk in each group. And the vaccine efficacy, you can think of this as 1 minus the relative risk. We haven't eliminated any infections. The bottom row of this corresponds to that 75%. So 1% infections in the placebo group. We're down to 1/4% in the vaccine group. Our relative risk is 0.25. And now 1 minus that relative risk, 0.75, we've eliminated 75% of infections. So we want the vaccine efficacy, we want VE to be 100%, but obviously that's not going to happen in practice. We are requiring statistically to show we are better than 30%, and that will require us to observe about 50% in these trials. All right, so where might that 50% come from? And this is all back of the envelope. Keep in mind that current estimates of the native reproductive number of this virus are typically above 2, might be higher than that. I'm not going to get into all the estimation involved with R, but remember, to actually reduce spread, we need to have R less than 1. So if we're talking about an R above 2, then, if R is equal to 2, we have to eliminate 50% of infections to get R less than 1. And in addition, note, not everyone was going to take this vaccine. So certainly we'd like it to be the higher the better, but you can start to see why a vaccine efficacy of, say, 20%, even though you're preventing some infections, that's probably not going to be enough to really eliminate the spread of this. And 50% is kind of a borderline case. We obviously are looking for a little bit better than that. But this gets an idea about why we're really looking for a decently high as opposed to just, are we effective at all? All right, so let's get into these trials. The overall sample sizes differ, but they're all in the tens of thousands. Moderna and AstraZeneca are about 30,000. Pfizer started at 30,000 and went up to 44,000. And Janssen goes to 60,000. So all of these are quite large. Randomization is 1-to-1 in all trials except AstraZeneca, which is doing 2-to-1 preferential to treatment. 2-to-1's just a sponsor choice here. You'll get more patients on the vaccine arm in terms of safety. You might take a little bit of a power hit by doing this, but certainly all of this is standard ratios. All of these are trying to show superiority to 30%. Statistically, again, we need observed vaccine efficacy in the 50% range to do this. The maximal events, so remember, we're waiting for infections in the population. All of these come out in the 150 range. Pfizer's is a little bit bigger for reasons I'll talk about later. And notice there's a question mark on Janssen. They're aiming for 154 as the max, but there's a quirk in Janssen, which I will get to. I think a good quirk. So certainly don't want to indicate that that question mark is questioning the study. It's just they have an interesting aspect on how they do their interim analyses. All of these are powered for vaccine efficacies in the 50 to 60% range. AstraZeneca has powered for 50%. And I'm not going to spend a ton of time on the analyses, but they all have differential kinds of analyses. AstraZeneca is doing a Poisson regression. They're including age as a covariate. This is allowing some of the soak up some of the variability. The Janssen and Pfizer trials are really doing binomials based on how the events split between the arms, and Moderna is doing a Cox proportional hazards. All of this said, the real driver of whether you're going to be successful is going to be the event split. There's nuances here, but the real thing that you need is more events on the placebo arm than you're seeing on the vaccinated arm, on the treatment arm. So these trials are all in a very similar vein. To get an idea about where this sample size comes from, what we're trying to do is we don't want to wait an incredibly long amount of time for events. So in the US, where I live, the current new daily cases rate is about 40,000 a day. In 100 days, that would be 4 million cases, which is about 1.25% of the population. The actual we know we're not testing everyone, so maybe the actual events in a very surveilled population is going to be higher. But about 1.25% of the population, with 30,000 patients, that's 15,000 per arm. And so 1.25%, just taking this, of 15,000 placebos is 187 events. So you can see what we're really trying to do here is make sure in a reasonable amount of time, we're going to see those 150 events. So this is likely to be within 100 days, less than that. So one of the reasons we're enrolling these tens of thousands of subjects is to make sure we expeditiously see events and can draw a conclusion. All of these trials are being monitored for a variety of adverse events. Some of the trials have very explicit futility rules. If we see these kind of events, we're going to stop. All of them have a DSMB. And the DSMB is empowered to recommend termination of the trial for safety concerns. So they're all being monitored for events. They can also all be stopped if there's a problem. The kind of events that we're looking for is, we can talk about shorter term events. So this is reactions to the injections, reactions in the days to weeks, small number of months range. And in terms of this, the sample sizes are quite large. So we're vaccinating 15,000 people with the active arm. So we're going to get a sample size in that 15,000 range on adverse events in the short term. So these kind of events we're likely to estimate well. We're going to get a really good idea of what happens in, say, the first week after injection, the first month. Longer term follow-up, remember, we're running these trials in order to get an answer within this small number of months. I just did the calculation with 100 days. So we're not, at the time of an emergency use authorization, we're not going to have a year of follow-up on these patients. Now, most people view that there are relatively rare events with long-term follow-up. So that's why that emergency use authorization is being considered, is that these long-term follow-up events are not considered to be big enough to limit use of the vaccine. But in terms of eventual approval for a vaccine, we're going to need to see this long-term safety. So it's a key question on how much long-term safety we need. For those following the news, there's been some discussion of requiring 2 months of follow-up for either 50% or 100% of the subjects. That seems to have been, at least in the US and the FDA, that seems to have become a very political issue. But in any case, this kind of long-term follow-up, this is the real crucial discussion on safety. How long does it need to be? How many patients do we need to see with a particular amount of time? If you're going to require 6 months of follow-up for everybody, it's going to take a long time to get to a vaccine. If you're only going to require 1 month of follow-up, then you only have 1 month of follow-up. So this is what medical experts, what I'm certainly looking for my own family, is to get an idea about what is appropriate and what's safe. The Janssen protocol is explicitly requiring 50% follow-up for 2 months for at least half the, or sorry, I said that wrong. The Janssen protocol is requiring 2 months of follow-up for 50% of the subjects. So that's long-term. We're also interested in disease severity. In many cases, that's an aspect of efficacy. So certainly, like the flu vaccine, they often tell you, even if it doesn't prevent disease, it may prevent severe disease. That may certainly happen with COVID. Paradoxically, some vaccines can actually increase disease severity. That may not be likely, but it's something they are monitoring for. And in terms of this, we're not going to get a ton of data. So remember, 150 to 165 events, severe or critical disease is a subset of that. So the sample size is going to be smaller. We are likely to get trends, but not conclusive evidence of disease severity, certainly not quickly. As the trial continues, this kind of information will be filled in. And it's important to note here that even once these event triggers are reached, these trials are all going to continue. So I certainly hope that we continue to see this follow-up and it's transparently given to us in real time as these trials continue, so that we can continue to make informed decisions about ongoing vaccination programs. All right, so talk a lot. So certainly I'm a trial designer and a statistician. So the interim analyses are interesting to me, and they've also been an aspect of a lot of the news articles. All 4 of these trials have interim analyses. What those allow is at certain points, efficacy can be declared prior to the maximal event count. AstraZeneca, Moderna, and Pfizer are all basing these analyses on event counts. And in the next slide, I have a detailed prescription. Janssen's are actually a bit different. So Janssen's are going to happen more continuously, at least weekly, but they require certain conditions to start. And so this is an important aspect which we will get to later. It's important to note, and the details are a little bit later, all four of these plans, so all four trials, protect against false claims. So these interim analyses, they've all been set up to make sure that they have a 2.5% chance of type 1 error. And what that means is if a vaccine is truly only 30% effective, remember, we want to show we're better than 30%. If a vaccine were only 30% effective, then each of these trials only has a 2.5% total chance of falsely declaring efficacy. So that's the total across all the interims. The trials are all obtaining 85 to 90% power. And power says suppose the vaccine was actually effective, either 50% for AstraZeneca or 60% for the others, then there's an 85 to 90% chance we detect that. So we certainly don't want to spend all this effort and miss an effective vaccine. So all four of these plans are limiting our chance of error. We're not going to declare an ineffective vaccine effective. We're not going to miss an effective one with these probabilities, 2.5% and these 85 to 90% power. All right, so here are the details of the plans. So remember the maximal events all in the 150 to 165 range. AstraZeneca is doing an interim at 75 when half the events are in. Moderna is doing interims a little earlier, one at 53, one at 106. Pfizer is doing 4 interims starting at 32, 62, 92, and 120. And what Janssen is doing, let me kind of skip to the bottom of this slide. Janssen has conditions on when the interims start. So they need 50% of the patients to have 2 months of follow-up. This is that condition we talked about at the few slides ago. And they're looking for 20 COVID events. So notice that's lower than Pfizer, but also keep in mind they're looking for splits here. So they need at least 5 severe, and they need at least 6 that are greater than or equal to 60 years old. Given I actually don't know what their enrollment's going to be. I don't think anyone does. But I would imagine since they have 60,000 patients, the real driver of this is going to be the 50% of patients with 2 months of follow-up. So it may be quite a few events before Janssen will start. However, after Janssen starts, they're going to do interims weekly. And then they're going to go up to their maximum of 154. If it takes more than 154 events for Janssen to hit this condition, so they have, say, when 50% of their patients have 2 months of follow-up, if we already have 154 events, then Janssen's just going to do a final analysis when that condition is met. Okay, so that's when these interims are. All of these trials have a variation of what's called alpha spending. So they are looking for certain conditions to be met at the interim. So AstraZeneca, when they do the 75 interim, what's going to happen is they need a p-value that's less than 0.00155 in order to declare efficacy. If they get that condition, they'll declare efficacy and presumably pursue the emergency use authorization, as long as they have a sufficient safety database. If they don't get it at 75, then they're going to go to 150, and then they need the p-value less than 0.0245. So again, combined, these two conditions maintain that overall false discovery rate. Or actually, that should be type 1 error rate here. So Janssen is basing theirs on something called a sequential probability ratio test. It's a little more complicated to get into on these slides. But they also control type 1 error. Moderna, you can see their p-values required, and Pfizer is doing a Bayesian analysis where they are looking for the posterior probability that the vaccine efficacy is greater than 30%. So this is what they need to show. Same hypotheses as the other trials, but they need that probability to be greater than 99.5 at the interims and greater than 98.6% if they go to 164. And again, those numbers have been set. So the 99.5 is presumably a sponsor choice. The 98.6, they've calculated what they need, and I verified this, everything looks good, to get a 2.5% overall type 1 error. Okay, so that is how the interims are set up. This has been controversial at times, certainly on Twitter and some news articles. Declaring efficacy is often called stopping early. I think this is a little bit unfortunate for reasons I'll get to in slides. It implies there's on time, there's something magic that happens at 150 to 165 events, and I'm not sure the magic is what people often think it is. These are all standard methodologies, so certainly similar trial designs have been improved by FDA, other regulators for decades. Even the Bayesian design at this point is an old design. These are all well within the current regulatory guidance. However, it is important to note that these are new. Certainly Janssen and Pfizer, they're among the first for vaccines that allow greater than 2 interims. So certainly this is kind of a distinction to statisticians. These would often be considered common, well-understood designs. But again, if you work in different clinical areas, you may have seen these, you may have not. Okay, so let's talk about what it means to stop early. So the advantage to these, certainly looking earlier, gives you the chance to get an effective vaccine to market faster. If one of these were 90% effective, where our deaths are certainly in the US are 500 to 1,000 a day. Getting a vaccine to market faster is wonderful. But they've also been criticized as shortcuts. So I want to talk about what we ought to worry about, what shouldn't we worry about. And I want to consider this for both efficacy and safety. And I want to bring back to a client discussion I had once, really interesting, smart client. And we were talking about a needed N equal 200 sample size. And they got into, they had done the calculations before, and we were really into, what does that mean? Why do I need N equal 200? And it became clear during the conversation that they had it in their mind, and I think it's reasonable to kind of think through it this way. We often talk about, you need this N equal 200. And they had it in their thought that the evidence just got bigger and bigger as the trial went on. And so what happened is kind of something magical happened at 200 where, this p-value goes below 2.5%, and that's likely to happen, not between 199 at 201, I guess that's a little facetious, but right around the N equal 200 spot. That's when we get sufficient evidence. And unfortunately, that's not really why we're picking an N equal 200. that doesn't reflect the time we have strong evidence. It reflects something a little bit different on how we're protecting ourselves against risk. And so let's talk about this. And I've said a lot of what's on this slide, but let's focus on the Janssen-Pfizer analysis. I'm doing this because it's a little bit simpler for me to graph. And the Janssen-Pfizer analysis, that's based on the event split at the time of analysis. So what they're going to do is, if we're doing an interim at, say, one of the interims for Pfizer is 120 events. If the vaccine had no efficacy whatsoever, it was 0%. This is way worse than we want. But at 120 events, the vaccine's doing nothing. So we should expect half the events in the placebo arm. We should expect half the events on the experimental vaccine arm. At 30% effective, then the vaccine arm should have 70% of the placebo arm events. So remember, we're getting rid of 30%. So the proportion of events on the vaccine arm, we've got 0.7 on vaccine, and then remember, we've got a full 100% on control. So the proportion on vaccine is going to be this 0.7 over 1 plus 0.7, and so that's 0.4118. So if the vaccine is 30% effective, when we're looking at the split, we should see about 41% of them on vaccine, a little bit less than 50-50. Obviously, as the vaccine gets better and better, we expect fewer and fewer events on vaccine. If the vaccine were 100% effective, we would expect 0 events on the vaccine arm. So what we need to show here is that the true rate is less than 41.18%. That's the hypothesis, formal hypothesis here for Janssen and Pfizer. So how would a 30% VE vaccine behave? So let's kind of think through this for a minute. We know it would produce 41.18% on the vaccine arm in the long run, but that's subject to variability. So it might be more or less as the trial evolves. And I know statisticians, this is often old hat, but bear with me, I'm trying to make what I hope is a cool graph to kind of illustrate the interims. So one possibility here, if we look at the events over time, so we can think about what happens when we've seen 1 event, 2 events, 3 events, 4 events, and all the way up. I've taken, remember these trials, the maximal events are about 150. I've taken this all the way up to 200 events. So this is one possible path. And so I've drawn this 0.4118. So this is kind of the goal. And we want to be below this. So we want to be below the line. Good vaccines are down here. A vaccine that's actually 30%, we expect it to oscillate pretty wildly at first and then stabilize around this line. But keep in mind there's variability. So this is another possibility. I generated random numbers on my computer. In an actual trial, we could expect anything along these lines. Again, variability. This one actually ends up a little bit lower here, and that's just standard variability. A 30% VE vaccine can just get a little bit lucky and come in a little bit lower. So let's think about the pattern of variability that we would see. So we'll generate 100 such paths. So this is 100. By the way, I really like the artistic merit in these little lines here. The way these kind of make this little grid. But anyway, you can see the variability. It's large at first, and then these start to stabilize, but they're not exactly 41.18%. So there's certainly still some variability here. So some variability exists at 150 to 200 events. In order to make a claim of efficacy, we don't want to mistakenly conclude a 30% VE vaccine works. So we're only going to conclude efficacy if we can get a result below this range of variability. So we have to have data down in this area below the black paths. And formally what we do is we look at the 2.5th percentile of these black paths. So I've got 100 lines here, kind of chop off the bottom two, and we do the math to make sure we actually, get the right rates here, not just random data. But so we need really, the data needs to be below these black curves. If we get that, we'll declare success. We are protecting ourselves against bad luck. And here, that bad luck is a dud vaccine happens to get a little bit lucky. We don't want to conclude efficacy in that case because we might make a mistake. We might conclude a dud vaccine is actually effective. So we need to be below the black curves. All right, so let's think about how a 60% VE vaccine would behave. So now I've added green paths onto this. In the long run, what we expect is a 60% vaccine efficacy. We eliminate 60% of the infections. We're left with 40% compared to the 100 on placebo. And so we expect 28.57 events to be on the vaccine arm in the long run. So these green curves are starting to cluster around 28.5%. You can also see it's variable how we get there. Early, there is a lot of overlap between the black and the green. And so the reason that we need 150 events, what we're waiting for is we're waiting for the black and green paths to separate. And that's happening sufficiently about 150 events. Back earlier, there's a lot of overlap. There's a lot of dud vaccines that look kind of like an effective one, and the effective vaccines can get unlucky and look a little closer to a dud. So, there's a lot of overlap here. That's why we're waiting. Out here, they're almost fully separated. Not completely, but they're getting almost fully separated. So when we talk about the discussion, all of this discussion about stopping early is somehow implying that 150 events is the right time. And I want to emphasize that it's not the right time as that's when the evidence finally accrues. It's the time when these have separated. If we're only going to look once, what we're trying to do is simultaneously control our false discovery rate. So we're waiting for wherever we declare success, we want all the black curves above it, all the duds, and we also want most of the green curves below it. And so you can see we've got these percentiles. Our cutoff for efficacy, declaring efficacy, 2.5 percentile of the black, and we're doing the 90th percentile of the green. So that's where the 2.5 and the 90% come into play. So we're waiting for this to separate. We're worried about bad luck in terms of a bad vaccine looking good. We're protecting ourselves against bad luck of a good vaccine having kind of a rough start. We don't want to miss a good one. So if you can look only once, if you're in this paradigm where I'm only going to look once, you want to wait for this. And so you are waiting a long time for this separation to occur. And so you can think of this as an insurance policy. I'm protecting myself against the bad luck, a dud vaccine looking good, protecting myself against the bad luck of a good vaccine looking bad. So this takes a long time. This insurance policy is very expensive. So 90% powered trials, we're really waiting a long time. So it's not clear that 150 is on time. It's when this separation occurs. The motivation for interim analyses is that If you look at this, there's a lot of these green paths where the data are firmly below the black. So these green paths down here, none of the black curves are in this range. So now not all the green curves are down there. So we didn't want to just only look here because of the overlap, but some of these green paths are sufficiently separated that in effect we know they're green. They're below all the black. They're good vaccines. There are some here, some here. So all of this area before we get to 150, there's plenty of green paths that have sufficiently separated. So this gets to the notion that we don't know when convincing evidence is going to arise. That's just the randomness of the process.
Kert, can you just say a little bit about time on this graph? Have you got some kind of assumption here about how long it takes to see events, to see infections?
So I certainly don't on the graph. Again, what we're expecting here for these trials is that this is in the small number of months range. And also keep in mind that accrual may start. So as patients accrue, events should start happening faster and faster. So early in the trial, you may have only vaccinated 5,000 people. The events are going to accrue slower. After you've vaccinated 30,000, you have a lot more opportunities. That's the wrong word, but anyway, there are a lot more chances for people to be exposed because there are more people. So time really kind of accelerates on this graph.
Yeah. Okay. So we've got the x-axis is number of events, and initially they will come in quite slowly and then get faster and faster. So, yeah.
Correct.
Okay. Thank you.
Yeah. So I would imagine that as you get out to 150 and 200, this is also the advantage of that longer term follow-up. Even after an EUA, we should continue to see this. And so we may see hundreds of events going into, say, summer of 2021, which is a lot more data than we're going to have now. What I'm getting at now is there are certainly some instances where the data is convincing earlier than 150. If the vaccine efficacy, if we were lucky enough to get an 80% vaccine efficacy, then that's the blue curves. So you can see they actually do separate very quickly. So we can detect that almost immediately. All right, so this interim analysis is to keep in mind that this maximal event size is protection against bad luck. If bad luck doesn't occur, we're going to find out the answer sooner. And the interims are designed to make a decision when it's clear. And I'm focused on declaring efficacy, but I should go back a slide. Keep in mind, there are plenty of situations where we should also declare futility. And all of these trials have provisions for futility where it's clear we are in the black, we are not in the green. So there are lots of reasons to also do the reverse. So these trials could end early for lack of efficacy. Okay, so I want to talk about what the Pfizer has this nice table in their protocol. This is, I think, in section 9.5, if any of you have it open. But this is giving the interim analysis plan and the boundaries for efficacy and futility. So Pfizer is doing an interim at 32 cases, and they need a split of 6 out of those 32. So a 6-26 split. If they get this or better, they get a declaration of success, declare efficacy. If it's 15-17 or worse, they're going to declare futility. If it's in between, then they're going to go to 62 cases. And you can see the required split: 15 or worse, 26 15 or better, 26 or worse. If that doesn't happen, 92, you can see the split: 25, 67 or better. You could stop for futility. At 120, there's a possible split. There's no futility boundary, and I think this is related to the question that was just mentioned, that the events are starting to come in relatively quickly. Also, declaring futility may have less meaning if they expect to be fully enrolled by this time, because all the patients are going to be followed regardless. So there's no more interventions with the subjects. And then finally, at the final analysis, 164, and you can see the 53 split here as well.
Kurt, can you just, so those splits between events, can you just say something about the events? So they, have the companies all defined event in the same way? Do we know? Is it just any test for positive?
They have not. Let me actually switch a little bit. I'll talk about this in a moment. There are differences here, so we expect them to be different for the different companies. AstraZeneca is looking for PCR confirmations 15 days after the second dose, second vaccine. So if you get COVID 2 days after the first injection, then you're not in this population. But once you get 15 days after the second dose, they're looking for PCR positive tests. Pfizer is looking for NAAT confirmed tests. Moderna is looking for PCR, but also symptoms. And Janssen is actually looking for moderate or worse confirmed illness. So these are all a little bit different. And Janssen is looking for, I think, the most restrictive definition of a case here. And I don't mean restrictive in a bad sense, just how long these are going to take. So they are different. And there's been discussion on Twitter about the different endpoints here, which I'm not going to go into in a lot of detail.
And
is that what you were looking for?
Yeah, I think so. Yes. And sort of on a similar note, but will these trials give us any sense of how long the protection lasts?
That's a good question. So let me table that for a moment and come back to it. So the answer is yes, but maybe not early. So these are the Pfizer. So this is the table here. And what I've done is I've plotted those regions where Pfizer will declare success. So if you can get into the red rectangles, so this is at 32 events, and they need to have 6 out of 26 or better. So if the path goes into this range, Pfizer declares success. At the next interim, 62, it's this range. At 92, 120, and the final analysis at 164. So what these are aimed at is all of these red rectangles are below the black region. So we're trying to grab, we know we can't be definitive at 32, there's too much overlap, but sometimes we can get a definitive answer. And I haven't graphed the futility, but sometimes we know we're in the black region, we've got a dud. Sometimes we know we're underneath the black region. We are no, and no means relative to this type 1 error rate, but we know we're effective. So this is what these interim analyses are trying to do, is if you're on one of these paths that separate early, then earlier than 164, we're trying to pick them up. All right, very important point here, and then I would definitely want to move to the safety issues. You do need to take into account the multiple looks when you're computing the type 1 error rate. There are lots of formulas to handle this. Each of these trials actually does something a little bit different, but they do all control the rate at 2.5%, and this accounts for the multiple looks. So you can see that they're less than 2.5% at each look. They're 2.5% in total. And so they've done this by Land-Metz for AstraZeneca, this sequential probability ratio test for Janssen. Moderna does O'Brien-Fleming, and Pfizer is doing basically a sum over the binomial probabilities, which is the standard Bayesian version to get this, the Bayesian hybrid version. All right, so essentially for efficacy, we should have an idea about whether we have a 30% VE vaccine or not. We should be confident of that. So let's get into this safety, and let me talk about the durability of immunity at the same time. What's stopping at 32 or 53 or 62 or 75 or all of these other things does is it does limit the amount of available safety data. It doesn't really limit the short-term injection reaction kind of events. So we're going to see thousands of patients get injected. We're going to see thousands of patients for the first few weeks. So that's definitely going to happen. So even with 32 events, those kind of sample sizes ought to be large. For long-term follow-up, this gets back to the question of the timing of the stop. If Moderna stops, for example, and I'm just randomly picking trials here, if they stop at 53 events, when does that occur? And so it depends strongly on the overall COVID incidence. So that could be quick, could be slow, depending on how many events they're seeing. If they enroll in an area where they have high incidence, then these events could happen quicker. And so the real issue here is how much long-term follow-up are we going to have? And it's unclear what is sufficient. So a lot of the debate that you're having here is, do we need this 50% of patients followed up 2 months? Do we need all of them followed up 2 months? And again, I'm not an MD. I don't want to wade into what is sufficient, but when you're paying attention to this debate, you should be looking to medical experts, regulators, everybody else on what is the right amount of follow-up here. One thing to keep in mind is the diminishing returns of this. So if you double a sample size, you only reduce the accuracy of estimates by you only increase accuracy by 30%. So do keep in mind, if we're talking about, say, some adverse event, and we're estimating it at 8% plus or minus 3.5, if we doubled the amount of follow-up, the amount of person years that we have, that might become 8% plus or minus 2.5%. So doubling that sample size, doubling the information doesn't necessarily help us. So we don't want to reflexively go, oh, let's double, triple the amount of follow-up. The amount of return, you need to have a good idea what is the real needed amount of information. And I certainly am not qualified to say what that is. But it would be nice if people would quantify this so that we could have a good idea. So this is going to be a concern with the early stops. Not that they're making false conclusions on efficacy, but they have insufficient follow-up. There's also concerns about subgroups, particularly at-risk subgroups. And keep in mind that all trials are underpowered for subgroups, and you're going to have the same issue. Doubling the sample size is only going to increase the accuracy of estimates by 30%. Is this valuable? That's not rhetorical. This is something we should decide. So this is something that the regulators are going to have to decide in approving these vaccines. All right, so I don't have a slide on durability, but it's a very important question. You have the issue that suppose the vaccine was only efficacious for a short period of time. The early events are going to be in that short amount of time. So you do have the possibility that if immunity wanes, later in the trial, you're going to see this effect attenuate. So that's certainly something, if we're talking about giving an emergency use authorization, we may want to pay a lot of attention to this follow-up and make sure that that durability is there, that this effect is not disappearing. Now, certainly a flu vaccine we give every year, that's a possibility for COVID. So even if it attenuates, but it lasts many, many months, that may be sufficient for really, stopping the pandemic.
Sorry, while you're on, just passing on another question. While you're on the subgroups,
I'm about done. So this is great.
Okay. Do any of the trials do anything to address, ensuring they're covering, different populations, different races, different genders, different age groups?
Yes.
Are they stratifying? What are they doing?
So they're all stratifying on risk to some degree. The details are in the protocol, but stratifying on age, risk categories, they're all doing various things with that. And it all seems perfectly reasonable to me how they're doing it. They all have some discussion of how many at risk. So there's a discussion of having a sufficient proportion of elderly people, sufficient proportions. One of the reasons Pfizer increased their sample size was to recruit different subpopulations. So they're all there. I want to emphasize that Janssen's requirements also act to constrain the interims. So Janssen allows us to stop really early, but they really won't start until they feel they have enough events and enough in subpopulations and so on. So certainly if the FDA were to say suddenly we have these requirements on a vaccine, the natural thing for, say, Pfizer to do is to say, well, we won't look at 32 because we're not going to have a sufficient database at that time. So it would be good to have this kind of guidance, recognizing that seems to be difficult at this time.
Thank you.
So anyway, my summary here, I know we're getting close to the top of the hour, is that all of these trials are well within standard practice. There is some novelty within the vaccine space for the trials with more than 2 interims. All of them protect against false claims of efficacy. Some of these allow efficacy claims more quickly than others. For most of these trials, a key issue is going to be the safety database. These early stops, we're not making false efficacy conclusions, but we have to be concerned about the safety database and making sure that that's long enough. So thank you. Happy to take questions. I don't have a hard stop at the top of the hour. So happy to talk for a little while.
Okay, I've just got one other question that has been typed in on the chat. And that was, this is a slightly technical question. Are we talking about false discovery rate as opposed to family-wise error rate? And would that make a difference?
We are not talking about false discovery rate. I did write it incorrectly on one of the slides. So these are all type 1 error. Their false discovery rate usually applies to so within the interim analyses, it's the family of interims. So at 2.5% total across making any mistake at any interim. There are no restrictions across the 4 trials. So each of them has their own individual 2.5% type 1 error.
Oh, I've just seen we're getting a few more questions in. Was there any consideration between the companies that they could share control?
I certainly wish they had. We are going to enroll an awful lot of controls here. So let's go back and do a quick tally of this. Where is my sample sizes? All right, so here is 15,000 controls. We're up to 45,000 controls, 60,000 controls, 82,000 controls. That's an awful lot of information about controls. It's not particularly efficient. Now, keep in mind that would have required discussions on matching the companies would have had to work together and do that quickly, but certainly these trials could have been faster, could have been more efficient, and gotten to a larger number of events sooner had they shared controls, yes.
Okay, thank you.
Not without challenges, but certainly the basis of platform trials these days. I'm sorry, Tom, let me mention one other thing.
Yeah.
It's important to note that a lot of the treatment trials are sharing controls, and that's been exceptionally valuable for getting treatments. And in fact, we've seen treatment decisions long before the vaccines are going to read out.
Should we read anything into people being invited back for the booster? Is that just a fixed part of the protocol? They come into the trial, they get their vaccine or the placebo, and then the protocol says 2 weeks later or a month later they come back and get their booster? Or is the booster related to interims or some kind of adaptation?
So yeah, it's not related to the analysis. So AstraZeneca, Moderna, and Pfizer are 2 shots by design, about a month apart. I think there might be a slight difference between them, but they're about a month apart, a little less. Janssen is a single shot, which, you know, given my children, that gives them 1 chance of passing out rather than 2. But anyway, so Janssen is 1, but the others, it's fully per protocol intended to be 2, and it would be 2 in clinical practice.
An interesting question. Why is the definition of 2 months follow-up based on the percentage of patients rather than the fixed end? Surely that 2 months of follow-up, we should be talking about, you know, we need this much data rather than, oh, you stopped early, we don't need so much follow-up.
I think that's true, actually. And so I think I agree. I think it's the 15,000. One possible concern about the Janssen trial, and it's not a concern about that it's not a concern about the negative to the trial, but waiting for 50% of this larger sample size may delay Janssen reading out. They will have more information when they do, but they may be later.
There's a question I can answer. I'm not sure whether we're planning to make the slides available, but we're certainly making the recording of this.
Yeah, the slides will be available.
Okay.
Well, we will do that.
So the recording will also be available, and we'll upload it to YouTube, and there will be a link to it from our website.
Yeah, and for those who follow me on Twitter, I will tweet out the link when it's set up.
Do we know, is there a minimum follow-up for all subjects? Will the
so all subjects are followed for 2 years in the study. There's no minimum in terms of when the emergency use authorization might be given. You know, they might have enrolled 25,000 patients, and the last few are fairly recent, and the first few have a lot of follow-up.
So Kert, this is Scott, question for you. So there's been a bit of, terminology like shortcuts and all that. You clearly mentioned you don't believe these interims are shortcuts at all. So if the result comes out relatively early at an interim analysis, in your mind, there is no shortcut to that. So you believe that the result is incredibly powerful. And would your opinion, maybe I've answered your question, but would your opinion change if it comes out at an interim as opposed to final?
No, my opinion about whether the vaccine is efficacious won't change. I will be looking at the safety database, I think, like everybody else, especially for the interim analyses where that follow-up was not an explicit part of the interim timing.
And have you calculated, the type 1 error is super superiority, so under the assumption that it's 30% effective. Have you calculated the type 1 error if in fact 0 is the truth?
Yes, not for all of them, but it is exceptionally small.
Could you, we have on that slide you're showing at the moment the different endpoint analyses. Do we know if those actually make much difference? Do we know, and it can be, is there any thoughts as to why they've picked different methods?
So all of these are basically standard methods. They all primarily hinge on the event split. A couple notes here. The way Pfizer is doing their interims, they are more likely to stop early. So, if you think about those red rectangles, the others are a little bit lower and Pfizer's are a little bit higher. They're all firmly out of the black, so to speak. But but Pfizer, and because Pfizer is giving them a better chance of stopping at the earlier interims, that's one of the reasons that Pfizer needs this 164.
Yeah.
So that's that, and that wasn't part of your question, but I wanted to make that point. But I think the binomial, the Cox proportional hazards are going to be about equal. AstraZeneca is including age as a covariate, so I don't think the Poisson regression is much different than the others, but including age as a covariate soaks up some of the variability. And I think that's one of the reasons that you can see their maximal events is lower, and they're also powering, they're able to do 2-to-1 randomization, and they're powering for a slightly lower effect size, which usually requires a bigger number of events. But I'm guessing, and I did not verify this, but based on experience, a lot of this is because they're including age as a covariate.
In this setting, would it have been possible to borrow historical data from, well, not that historical, but the amount of information we already have on infection rates that's going on all around us?
So as a lot of people know, I certainly am a proponent of historical borrowing, and certainly a proponent of being explicit about all the caveats with historical borrowing. Here is a case I would not borrow historically. One of the reasons is that the incidence rates are varying so much across locations and across time that I would just be worried that the historical data wouldn't be sufficiently on point. So here I actually would not borrow the historical data.
So I've got a technical question on the Janssen trial, because they've got they're doing this very frequent interim process, and the question is are they doing full sequential likelihood ratio testing? Can they look at the patent officers?
Well, so almost. So in their what they're as my read of this. They're aspiring to do the full sequential. In all of these trials, as an operational issue, it's likely just impossible to look, after every event. So they have specified in the protocol the interims will be at least weekly after they begin, and they might be more often than that. This full sequential method means that they could look after every event and still control their type 1 error. So, but they likely will not, given the operational aspects of that.
And what will happen if a trial reads out successfully at an interim, but then in the follow-up, the data moves against them, and they would on some metric they would no longer look successful?
So that is unlikely, except for the durability issue. So I can see that attenuating. It is, and I'm not sure that's viewed as likely by medical experts. I defer to certainly to the medical aspects of this. That is exceptionally rare for if you stop at an interim and then you continue to follow up, it's very rare that you would see it fall back that much. The interim analyses are set up that you need so large an effect that those paths do not go back into the black region. So you're just far below it. And while that's possible, that probability is probably, and I haven't done it for these, but I've done it for other trials, it's under 1%.
What do we think will happen if one of these trials becomes approved at an interim by the FDA? What will happen to will the others just give up, or will they all carry on hoping to also be successful? Will it change how successful they have to be?
It certainly might change how successful they have to be. I'm sure if one of them read out with 90% vaccine efficacy, that we would expect the others to match it. I think in terms of the one thing to keep in mind, that this is not a trial this is a trial where they're enrolling quickly and then doing a lot of follow-up. So it's quite possible by the time that an emergency use authorization is actually given, that all of these trials may be fully enrolled or nearing it. So what we're really talking about is the follow-up period, and that follow-up would not end just because an emergency use authorization has been given to another vaccine.
And what would you feel if 4 of the trials were unsuccessful? What would you think about if 3 of the trials were unsuccessful? What would you think about an efficacy claim in the 4th? Would you become more skeptical?
So I personally would. We tend not to handle approvals this way, but certainly I would imagine if you came in and 3 of the trials had failed and the last one came in with the proverbial p-value of 0.024 and was successful, yes, I would be a bit skeptical that perhaps it got lucky out of the 4. That would also work in reverse. If 3 of them worked and the last one came in with the proverbial, p equal one-sided 0.03, I'd probably be inclined to think it works too. So yes, I would be inclined to borrow a bit. Now keep in mind that these vaccines also have different mechanisms of action. They're different kinds of vaccines. So I would have some subtlety in that, looking at how the vaccines are actually structured and how much to weight that kind of borrowing amongst vaccines.
If a trial stops early at an interim for success, does that affect the uncertainty in our estimate of its VE? Does that estimate shrink as it goes later? So an early stop, it might look very good, but we'd also have higher uncertainty about it.
So yes, we certainly would have higher uncertainty. Now keep in mind that uncertainty is going to be in a higher range. So for example, Pfizer at 32 events, if you get 6-26 split, which is a win for Pfizer, then the observed vaccine efficacy is about 77%. The confidence interval runs from 40 to about 100. So it's very broad, but it's all in this above 30 range. If you went to the end in Pfizer and got 53 out of 164, then the observed vaccine efficacy is right over 50, and the confidence interval runs from, I think, about 30 to 70. So it's certainly true that you have a wider range the earlier you stop, but that range is shifted up. So it involves higher values. From a Bayesian perspective, if you look at the posterior distribution of 6 out of 26 versus 53 and 111, what you get is a relationship called stochastic dominance, which essentially says that the odds the vaccine is X effective is better for the 6 out of 26 or the 6-26 split. So I think you do see extra uncertainty, but all that uncertainty is on good, better vaccines. So that doesn't faze me as much. And I certainly would expect all of these companies to continue follow-up and narrow that down over time. But I wouldn't have concerns about being vaccinated on the basis of efficacy. I would, in fact, have hope I'm getting a really good vaccine. I just don't know how good.
And I think
again, let me be careful to say that I would also be looking at safety.
Yeah.
So I want to make sure that's explicit here, that the interims are protecting efficacy.
I think I've got last 2 questions. So one is, if a vaccine has become sufficiently successful to get the emergency approval, what happens to the subjects on the control arm? Do they get vaccinated, or do you have to keep them on placebo in order to have follow-up data on them to compare to your follow-up data on the vaccine?
So I think that's going to be a discussion. In most cases, they would be followed up. I don't know the medical aspects of mixing vaccines, whether that makes a difference. It may not make a difference at all. I have no idea. I'm not an MD. But I would assume there is some amount of follow-up, and then if one vaccine's effective and the one they're on is not, I would assume there is some discussion of crossing them over, but I don't know the details of how that would happen. You certainly want to follow them up long enough to see adverse events and no safety.
And last question, could you comment on if a company is doing quite frequent interims, then would it be better to actually say, well, we want you to see success at one interim, but also at the next interim?
So not really, for a couple reasons. One, these interims are going to be very, very correlated. So at some level, it's redundant. If you are successful at one, you're going to be right at the cusp on the next one. The other reason is that I'm going to give kind of a nerdy response. There's something called the sufficiency principle, which says that you shouldn't have to worry about the order of the events in order to make a decision. And looking at things, looking at 2 interims would violate that sufficiency principle. So it really doesn't make sense to do that.
Excuse me. Well, thank you very much, Kurt. That's been an excellent presentation. I hope we haven't exhausted you too much with the follow-up questions. I hope folks think that I've properly reflected the questions as they come in. There was a wonderful flood of questions as you finish. And just to repeat, the link to the recording will be on the website within the next week.
No, I appreciate everyone for coming. Very happy to have you.
Yes, thanks very much for attending, everybody, and so many of you staying on for the extra Q&As after the hour. So farewell, have a good weekend. Goodbye.